Alright, this little lesson is going to be on population genetics, so it's going to further play off the work that we talked about within the third unit. It's going to take a look at specifically how to calculate percentages in relation to dominant and recessive alleles, specifically to dominant genotypes, recessive genotypes, and heterozygous genotypes, and it's going to work through how to calculate those processes. And then we're also going to go into a bit of the terminology about how we look at speciation and populations and how populations come to be and different variations come through and how we get the different variety that we get within organisms through a look at population analysis. So the first thing that we are going to look at is microevolution. Now microevolution refers to a small scale changes in allele frequency brought about by mutation, natural selection, gene flow, and genetic drift. So meaning you have a set of individuals, now their DNA sequence might change through mutation, which creates something new, natural selection, so those who are better fit are going to carry on with their genetics, and those aren't, that will die off. Gene flow, how new genes come into the system along with genetic drift, and we'll go into more of a concrete definition of those terms. But just overall, how are we going to change individuals, so the allele frequency, so the genes that are in our population through these different ideas. Now our gene pool is the various alleles, so gene pool looking right here, is the various alleles of all the genes in all the members of a population. So if we look at our school right now, our gene pool is everybody within the building. Now the population, so definition right here, is all members of a single species occupying an area at the same time. So the population of our school, we'll say 830 students, and the gene pool is all the various alleles of all the individuals within our population. Now an interesting point here is that individuals themselves do not evolve, only populations do. So I mean an individual might have a new allelic frequency change somehow in relation to what we talked about microevolution right up here. However, it's the population over time that changes as a whole. Now evolution occurs when there is a change in the allele frequency in a gene pool. So evolution occurs when there is a change in the allele frequency in a gene pool. So meaning we will have changes over time of our whole population when the allele frequency right here can be changed through mutation, natural selection, gene flow, and genetic drift. Now variations in a gene pool. So gene mutations, these will produce new alleles, and this is through, changing the DNA sequence. So you might have just simply one sequence change, or you might have multiple sequence changes. And these can be for good, these can be for bad, these can be lethal. There's a variety of different means that we've talked about in the previous unit that can result of a gene mutation. Now a big one is crossing over at meiosis 1, and this produces a new combination set of alleles, and this is where you have your tetrads lined up. So I have my two tetrads lined up, and then in the process of meiosis 1, when they're just that close, I have this tetrad, and I have my other tetrad, and they're going to have that site of crossing over, and then they're going to exchange new information. Now independent assortment is my next idea here. Independent assortment at meiosis 1, so mixed maternal and paternal chromosomes in the gametes. So meaning when my chromosomes are lining up here, there's no set law that says this chromosome has to be on the left side from the maternal side, this chromosome from the paternal side has to be on the right side. They are completely random, and they can be any which way. And it could be this one now could be over here, and the siting crossing over could then be on this side. So our changes due to crossing over and independent assortment can greatly increase our genetic variability. Other one here, fertilization combines alleles from two different parents. That's a huge way to increase your variation within your gene pool. Completely by random mating. It's not so much random. You might choose to mate with somebody, or an animal chooses to mate with a certain other organism. But by just having two mating partners who have different allele frequencies and different genes, you are going to create new information. And then you can also have changes in the chromosome structure or number, and that's where we talked about trisomy disorders and monosomy disorders. So through the process of non-disjunction. So when we looked up here, this is, meiosis, and maybe one, or instead of my chromosomes completely both going opposite ways like they have here and here, both of them will go to this way. So then I'm going to end up with chromosome that's going to have trisomy with 3 and monosomy with 1, and that's going to further change and have variation within my gene pool. Now to look at this mathematically, what we are going to do is we're going to use the Hardy-Weinberg principle. Now the Hardy-Weinberg principle measures changes in allele frequencies in a gene pool. Therefore it can be used to demonstrate the evolution of a single trait, specifically of one thing that we are looking at. Now the genetic equilibrium, so looking at a few definitions right here, is the frequency of a given allele remains stable one generation after the next, and thus there is no evolution. So equilibrium, we're trying to keep things nice and similar, the same. So meaning genetic equilibrium, you have no change. And that's not always ideal. You do want to have changes, you do want to evolve as a species, you want to become better suited for the environment. Now, a few points that are extremely important to remember here will be the 5 conditions that must be met to maintain genetic equilibrium. So, in order to have no evolution, first one is that you can not have, there's no mutation, so you're not changing your DNA sequence. You have large populations, and therefore you have no genetic drift, so no new genetics coming in. Your populations are isolated, so no gene flow, so nothing flowing out, nothing flowing in. So think of something on an island. 4, there is no natural selection, so what you start with is what you end with. So no, the best individuals are not the only ones living. All individuals are going to live. It's going to be the same genetic information that's going to be passed on each time. And number 5, there should be no random mating. So you are specifically within your isolated population with the same individuals. There is no new individuals coming in involving in the mating rituals. Now calculating the Hardy-Weinberg principle. Now these are going to be extremely important for you to remember. So you are not given all this information on a diploma exam, so this slide here is vital that you will remember it all. Now, first one, little p. So little p right here represents the frequency of allele A. So what do we know about capital A's? We know that that is the dominant allele. So P represents the frequency of the dominant allele. Q represents the frequency, little a here, of the recessive allele. And from that, we know that P plus Q equals 1. So there's a small little formula, very easy to find. If you have P, you can easily find Q. Now to determine the genetic or the genotypic frequencies, so up here on these ones we were looking at the allelic frequencies. Now to determine the genotypic frequencies, so homozygous dominant here, heterozygous and homozygous recessive here, from the allelic frequencies, so again here and here, the genotypic frequencies must add up to 1. Therefore, p squared plus 2pq plus q squared equals 1. And this is the Hardy-Weinberg equation. Now it's used to determine genotypic frequencies from allelic frequencies. So a couple important things to find. If you have p up here, you could find p squared. You could also find q, which could help you find q squared. And then you could also, if you have p and q, you could also find 2pq right there. So it's all related together. This slide here just further shows exactly what each letter is represented by. So again, P here, so this one is the frequency of the dominant allele. Q is the frequency of the recessive allele. P squared is the frequency of the homozygous dominant genotype. Q squared is the frequency of the homozygous recessive genotype. And 2pq is the frequency of the heterozygous individuals. So because you have a P and a Q, and again up here, heterozygous, big letter, little letter. Now, to put this formula in an example, we have a question here. So about 70% of North Americans can taste the chemical PTC, and the remainder cannot. The ability to taste is determined by the dominant allele T, and the inability to taste is determined by the recessive allele T. If the population is assumed to be in Hardy-Weinberg equilibrium, what are the genotypic and allelic frequencies of the population? So first off, looking right now, what do we know? Important point is that the ability to taste is determined by the dominant allele T. So which means anybody who is homozygous dominant or heterozygous can taste. So 70% of those individuals can. However, we need to find specific genotypic frequencies, so we need to find our p squared and 2pq. We cannot just simply break this 70 apart. We have to look at the 30% of individuals, because it equals, that's 100%, which are going to be homozygous recessive. So just to show you how we work, if we look right here, since 70% of tasters, this says TT, and again, that's a little error, that should be little t, because they could be heterozygous or they could be homozygous dominant, so either or. So since 70 or 0.7 are tasters, then 0.3 must not be tasters. Again, here's our homozygous recessive individuals. Since q squared represents our homozygous recessive individuals, we can determine q by taking the square root. The square root of 0.3 is 0.55. So we have now our q number. P plus q equals 1. So now, since we have q here, we can determine p. So then p must equal 0.5. Because if we rearrange this formula here, we will get p, taking this number 0.55. So 0.55 plus 0.45 equals 1. So again, this 0.55 equals q, and this 0.45 equals p. Now, to determine the allelic frequencies, or the genotypic frequencies, we can take p squared, so our number 0.45 here, square root it, and we get 0.2. If we want to calculate our heterozygous individuals, 2pq, so 2 times 0.45, which is our value for p right here, 0.55, which is our value for q right here, we get 0.5. And then to determine our homozygous recessive individuals, if we take 0.55 again up here, square root it, we get 0.3. And again, the Hardy-Weinberg is p squared plus 2pq equals, plus q squared equals 1. If we add 0.2 plus 0.5 plus 0.3, we see that our answers check out because they equal 1. Alright, next example question that we have here is, an investigator has determined by inspection that 16% of the population has a continuous hairline, and that is a recessive. So right away, we know that they are talking about a genotype, because the genotype represents or helps represent the phenotype. So again, if they're showing something, we have a number here that represents a genotype. Again, what are the allelic and genotypic frequencies of this population? So to go on, if we look at this, we were given our homozygous recessive genotype. So we know now that we have q squared equals 16%, therefore 0.16 equals, q is equal to the square root of 0.16. So square rooting 0.16, we get an answer of 0.4. That is the frequency of the recessive allele. We put that in our formula to find p, so we rearrange the formula. 1 minus 0.4, which we got here for q, equals 0.6. So that's what p equals. It's the frequency of the dominant allele. To get p squared, we will square our answer for p, and we get 0.36. That's the frequency of the homozygous dominant genotype. 2pq is 0.4, which we got up here for q. We have q squared or p squared right there, and we get an answer of 0.48, and that is the frequency of the heterozygous genotype. And then Q squared is 0.4, which we got up here, which equals 0.16. If we put that in our formula again, we put 0.36 plus 0.48 plus 0.16. It checks out because it equals 1. So I'm just going to skip over this slide of giving you a couple examples. We can do further ones in class. Now, a couple bit of the talk here about evolutionary change, and specifically about mutations. So we know that mutations provide new alleles, therefore change the allelic frequency of the gene pool. Because, I mean, if we have our DNA sequence and I had an A and a T and a C and a G and another C and a G and a T and an A, and all of a sudden I decided that I needed to change this T and an A here, so I'm going to change that T and A. We are going to change them now to a C and a G. I've created a mutation. Now, some mutations may not be detected unless there's an environmental change. So meaning they have responded now differently, or the organism has responded differently to its environment. So whatever was changed within its genetic code has shown an outward expression to where it lives. Or maybe it's made, an example of giraffes. Was it a mutation that helped make their necks longer and they were more suited? So through time, what this slide overall is saying is that mutations can provide new alleles, therefore changing the allelic frequency in the gene pool, which may be passed on. What you see with genetic drift is it involves changes in allelic frequency due to chance. So, that's going over. So example here, we have 10 squirrels, there's 9 brown ones and 1 red one. If the red squirrel dies, this is a major impact on the gene pool because, it's not going to be passing on its genetic information anymore. Now, to give you a little more of some other definitions that affect genetic drift, we have the founder effect right here. The founder effect is when a few individuals found a colony, only a fraction of the total genetic diversity of the original gene pool is represented. So let's say within our school here, we have our population, and then we close off a classroom. So our Bio 30 classroom is closed off. We are not allowed to interact anymore. We only have a few individuals from our original gene pool that are represented there. Therefore, the actual genetic drift and the change that we could have in the variability majorly decreases. The next one that we have here is the bottleneck. This is drift due to decline in population size. So a small population that is forced to exist as a small population over time will experience a decline in allelic variability as well as a likely change in allelic frequency. So an example here is the Amish in Lancaster, Pennsylvania have a recessive allele causing dwarfism and a high proportion of polydactylism. So 1 in 14 compared to 1 in 1,000 in a regular population. Another example that you could see with a bottleneck here was we have the founder effect that I talked about in our classroom. We are completely boxed off from all individuals within the population of our school here. Now, due to our bottleneck, if we had to survive as just our classroom set of individuals, we would have a decrease in genetic variability over time due to the low amount of individuals that are bringing genetic variability within our room. So genetic drift here, just an example we have within this picture. So right up here, this first one, we have a few homozygous red dominant flowers. We have some heterozygous individuals here, and then we also have a homozygous recessive. Now, only 5 of 10 plants leave offspring. So the ones that are highlighted in the white here, these are the only ones that leave offspring. Now what you're going to see is a change in the genetic drift because we have an increase in our heterozygous individuals who are bringing the recessive allele in, and only 1 homozygous dominant individual right here. So what you can see now is a lot more homozygous recessive individuals within our population. Again, only 2 plants of the 10 leave offspring, and it's specifically these 2. You can see due to genetic drift, we get now everybody that is homozygous dominant with the trait. Now just to go back, we have, due to chance here, these two individuals say, now we have a whole different change that can happen. Next definition that we talked about is gene flow. This is the movement of alleles between populations by interbreeding, migration, dispersal of seeds. So just meaning, how does the genetic information, so the gametes of individuals, how do they get interdispersed and with one another? Do we have, breeding here, interbreeding here? Is migration happening? What's coming in, what's coming out? And how are the genes, really, it's the term really is kind of self-explanatory. How is our genetic information flowing in and out of a population? Non-random mating, this occurs when individuals pair up not by chance but according to genotype or phenotype. So maybe interbreeding, they want to specifically be with certain individuals, or they are, if you think within some organisms, within animals, they look for the bigger, stronger, faster, so they only target certain individuals. Within humans, we all have our own taste, so we target only certain individuals. But one of the things, if you have within a whole population, if you have non-random mating, you are going to have an increased proportion of homozygous recessive forms, especially recessive abnormalities, because if you are targeting only specific individuals, those recessive genes that are going to come together will be further amplified because you're only continually targeting the exact same gene pool. Natural selection, so those that best adapted to the environment survive and pass their traits on to their offspring. It's the strongest evolutionary force, so the strongest, the fittest, the fastest within the animal kingdom. With even the plant kingdom, those plants that are best suited for their environments, they're the ones that are going to pass on to their traits. And this is something that you talked about in Bio 20. It's, really changes the gene frequency because only the best are going to be passed on. Now, natural selection, just to kind of further go into it, is a process by which favorable heritable traits become more common in successive generations of a population of reproducing organisms, and unfavorable heritable traits become less common due to differential reproduction of genotypes. So, those individuals who are wanting to mate with certain individuals who have certain characteristics, that is more likely going to happen, and those who have the unfavorable traits aren't going to get their mates, and more than likely they are not then going to survive or have their genetic information passed on. Now, natural selection acts on phenotype. So this is a really important point. It's not that you can actually see the genotype or the allelic frequencies of an individual. You can only see the actual phenotype. So this here is really important. So natural selection acts on the phenotype or the observable characteristics of an organism, such that those individuals with favorable phenotypes are more likely to survive and reproduce with those than less favorable phenotypes. Now, the phenotype's genetic basis, so the genotype associated with the favorable phenotype, will increase in frequency over time, because again, you're only passing on the good genetics, or the genetics that are more suited for the environment. Over time, this process may result in adaptations that specialize organisms for particular ecological niches, and may eventually result in the emergence of a new species due to changes that may happen. So in other words, natural selection is a mechanism by which evolution may take place within a given population of organisms, and a major evolutionary force that causes changes in gene frequencies due to specific targeted attraction, especially the phenotype of one individual to another. Now, there are three general outcomes of natural selection. We have stabilizing selection. This favors intermediate variants and typically occurs in relatively stable environments. Stabilizing selection probably prevails most of the time in most populations. We have directional selection, and this shifts the overall makeup of the population by acting against individuals at one of the phenotypic extremes. We have diversifying selection, typically occurs when environmental conditions are varied in a way that favors individuals at both extremes of a phenotypic range, and this can lead to a polymorphism, so occurs when two or more clearly different phenotypes exist in the same population of a species. So in other words, the occurrence of more than one form, or a morph. And so we can see here we have stabilizing selection, we have directional selection, so stabilizing is staying within our evolved population is kind of staying within the middle here. Directional selection, this is again, this blue dotted line is our original population, and it is moving away. And then diversifying is moving to the extremes compared to the middle and the middle of the original population. Speciation, just to further talk about a couple of different terms here. So species is a group of interbreeding populations that share a gene pool and are reproductively isolated from other species. So, I mean, they can only mate with their own, with the same species. It's not like you're going to have, you know, a lion and an elephant, say. Now, reproductive isolation of gene pools occurs when mating is never attempted or offspring do not develop. And the reason for this is that there may be different mating habits. So right here you have different courtship patterns, you have seasonal differences in mating, and the inability of sperm to fertilize an egg. So reproductive isolation, I mean, you're not going to have certain species, you know, you might have certain types of dogs mate together, but you're not going to have a dog, say, with a cat or anything like that, because again, they're going to have different mating habits, different courtship patterns, seasonal differences, and a big one here, the inability of the sperm to fertilize the egg. Now, steps in speciation. So it occurs when one species gives rise to two species by reproductive isolation. Geographic isolation occurs when two populations are separated from each other by, say, a new river, a canal, or a highway, and they can't get each other. And enough variation occurs between the separated populations due to independent mutations, genetic drift, natural selection example, the two populations become reproductively isolated when the populations are reunited. So you have an example here of geographically isolated squirrels in the Grand Canyon. I mean, over time, they are geographically isolated, but over time they will become reproductively isolated because they've been separated for so long that if we go back to those 4 points, I mean, they're going to have different courtship patterns, different seasonal patterns in mating. The egg might not be able to meet with the sperm, and so on. Now, a real important point here is that speciation can occur quickly. So, I mean, through punctuated evolution. So if you remember from Bio 20, you had, you know, one species, and then all of a sudden, boom, a new species happened, and all of a sudden, boom, another new species happened. And this could be a virus. All of a sudden, there was a rapid change, and a completely new species develops from what the original, and then what we have here is different species that are going to be reproductively isolated from one another. And that is the end of this little video here. Big thing is really make sure you have a clear understanding of the Hardy-Weinberg and how to use that formula. And there's further examples within the website and with our note packs to further help your understanding of that.